Hello, welcome to the project video for the Phase 2 aspect of the cryopreservation of large volumes of hepatocytes senior design group. Before we give you the details of our system design, here's a little background of our project. There are over 120,000 patients waiting to receive organs in the United States. Organ shortage in society is linked to several different factors. However, a major inhibiting factor that has posed to be a severe challenge in organ transplantation is the problem of sustaining the viability of organs for a short period of transportation to the patient. The Charlotte Banks Initiative, located in UNC Charlotte, aims to accelerate the development of preservation technologies that will extend the storage time of valuable tissues and organs so that more lives can be saved. In order to preserve complex structures like tissues and organs, a process must be developed to preserve the primary structures known as cells. Our senior design group was tasked with the goal to develop a liquidus tracking system that is capable of delivering the vitrification solution through a single inlet-exit flow to alginate encapsulated hepatocytes at a specified temperature and concentration. This system can over time be used to develop a technological system for cryopreserving large volumes of tissues. The system is made up of 4 essential subsystems, which are fluidic subsystem, thermal subsystem, control subsystem, containment subsystem. These subsystems and their relations to the system as a whole will be thoroughly explained in the following slides.
Here is the overall setup of the thermal fluidic system that we optimized this semester. As you can see, we have the chiller on the left here that is used to cool the refrigeration fluid that runs through the outlet tube at the back of the chiller. The outlet tube goes to the heat tape that is used to heat the chiller fluid until it reaches the desired temperature and then flows through the housing unit. Up here we have the 2 peristaltic pumps. For demonstration purposes, we have filled them with water and food coloring, but for the actual experiment they'd be filled with PBS and LT-DEG, and they're used to mix the cryoprotectant agent, which is the LT-DEG, and the buffer solution, which is the PBS, mix them together, feed them through the bubble trap, and then through the housing unit, which houses our heat exchanger and cell chamber. Now I will further elaborate about the heat tape. We added this heat tape so that we have greater flexibility in modifying the temperature of the chiller fluid than we originally had with just the chiller. The chiller itself is not able to cool down our vitrification solution quick enough, but this heat tape will allow us to do that. The power supply here supplies the voltage to our heat tape, which is then modified by the MIDAC, and goes through a transistor circuit. For verification of our system and for the concentration-based temperature control, we have incorporated this data logger, which allows us to change between different thermocouples that are placed throughout our unit and determine whether or not those temperatures are the same as those which we desire or expect. As you can see, thermocouple 8 is the one that is located right here between the outlet tube and the heat tape. We have thermocouple number 6, which goes into the top of the housing unit and into the top of the cell chamber. We also have a thermocouple that goes into the chiller, as you can see here. It goes into the chiller bath and is placed next to the outlet tube of the chiller. As I said, we can use these thermocouples to verify the temperature measurements and compare them to what we expect, and we can use them to send temperature data to the LabVIEW program so that we can control the pump concentration accordingly. Here is an overview of our LabVIEW program that was developed for this project. First, we have the start switch, which turns the pumps on. You can input the maximum desired flow rate that will be split up between the 2 different pumps depending on the concentration. Here are the individual pump flow rate outputs. Then this box here allows you to input how many steps you want to go through during your run. The table below is updated based on the number of steps and allows the user to input the bounds for the temperature. So between 30 degrees and 5 degrees, these pumps are going to run at 50% concentration. Then our heat tape is controlled by these 3 boxes here. So we have our program set up for a 3-stage test, meaning that the heat tape is going to be supplied a certain voltage for a certain amount of time. Then it is going to switch to the other voltage for the next desired amount of time, and then the last voltage until the end of the program. Now I'm going to show you the control we have over the pumps. I'm going to introduce a concentration to the LabVIEW program. Then I'm going to come over here and verify that we are in the temperature range, and you can see that the concentration is changed while we're in that temperature range. As you can see, we're mixing yellow and blue food coloring, and the output is green, showing they are mixing through the bubble trap. To run the LabVIEW program and control the pump's concentration, I'm first going to have the flow rate set to 5. Then I'm going to set the concentration to 0%. Then I'm going to click Start. You can see the change in the graph switch to the concentration that is being output, which is 0. And this temperature is the thermocouple temperature inside the cell chamber. Here you see that it is 12.2 degrees Celsius, and that 12.2 is perfectly between 30 degrees and 5 degrees. So we have a concentration output of 0%. And as you can see, 0% means that no LT-DEG is being pumped into the system and that only the PBS pump is running. If we take a look at the bubble trap, you can see here that there's a yellow drop dropping down, but we see no blue droplets, indicating that this is only PBS. If we return to our LabVIEW program and want to change the concentration that is being delivered by the pumps, we can change this number to 25 and immediately see that there's a jump in the concentration graph. We can again see that this happens in between the temperature range of 30 degrees and 5 degrees Celsius. And that is why the concentration is able to be changed and the pumps responded accordingly. While the device is being used to load and unload the vitrification solution, the concentration will change automatically in response to the temperature of the fluid entering the cell chamber. If you want to increase the concentration to 50%, you can see that there is a jump in the concentration curve. And if we go here to the pumps, you can see that they are both flowing at the exact same rate. And if we show the bubble trap, we can see that the blue and yellow drops are falling at the same rate from the trap. Let's say that you would like to increase the concentration one more time to 100%. You see the steep increase in the concentration curve and verify that the cell chamber temperature is still between 30 degrees and 5 degrees Celsius. And our pumps here, you see that PBS pump is not pumping at all, and we have 100% from this pump here, the LT-DEG pump. Now I want to show you the capability we have with our heat tape. As you can see, these boxes here represent the voltage that is applied to the heat tape. So when I click Start, the program starts at 4 volts applied in this demonstration for 1 minute, then applies 2 volts for 1 minute, and 0 volts for 1 minute. We're going to watch the thermocouple temperature right here, that is between the outlet tube of the chiller and the heat tape. We see that after 1 minute, the thermocouple reads a temperature of approximately 10.8 degrees Celsius, and we see that after this minute, the temperature is decreasing due to the change in voltage from 4 volts to 2 volts. After the second minute, the temperature of the thermocouple is approximately 10.3 degrees Celsius, and it is still going down because the voltage is now changing from 2 volts to 0 volts. After the last minute, we see that the temperature for the thermocouple decreases in approximately 9.8 degrees Celsius at the end. Now I will show you the inside of the housing unit. As you can see, it is connected here by a little clip, and in the back is connected by hinges. The housing unit is made of 2 pieces, which is filled with spray foam insulation that holds the heat exchanger in the cell chamber. This spray foam mold helps improve the heat transfer and keep the heat exchanger and cell chamber well protected from the warmer outside air. It also adds order to the system and allows experiments to be more repeatable. Here is the cell chamber. This cell chamber holds the alginate spheres that actually encapsulate the cells. It has a lid which has an embedded thermocouple. The cell chamber body has a diffuser which has these holes that diffuses the mixed liquid over the alginate spheres that sits on top of a mesh that holds the alginate encapsulated cells. This mesh can be removed and replaced when the spheres are inserted and taken out. Future work that could be done to improve the system includes adding a gasket between the top of the cell chamber and the body to keep air from coming in that space. The top of the cell chamber also needs to be reprinted to include the space for this gasket. Another improvement would be to flip the heat exchanger upside down so that the outlet tube of the chiller comes into the bottom of the housing unit. You really want all of the contact you can get from that fluid to touch the coil inside of the heat exchanger so that you can have optimal heat transfer from your heat exchanger to your vitrification solution.